Our group develops small antibodies called SCFBs as non-opiate treatment for chronic pain. The SCFB were recently optioned for licensing by Neurochronics. Our pipeline includes 2 small antibodies targeting receptors for CCKB and P2X4. Both antibodies deliver durable pain relief for over 3 weeks in chronic pain models, including nerve-injured and chemotherapy-induced neuropathic pain and inflammatory back pain, highlighting their multiple indication potential. An added benefit is that they prevent anxiety and depression that accompanies chronic pain. The 2 antibodies have been fully characterized and tested and are ready for IND-enabling studies. Our team has received significant external validation through $3 million of non-dilutive grant funding, with potential for additional NIH funding for IND-enabling and first-in-man phase 1 trials. The Neurochronics startup company is seeking further investment of $10 to $15 million currently toward the lead optimization and phased trials. Our goal is to develop non-opioid therapies for chronic pain. Easily recognizable is the unmet need for reduction of chronic pain, which is closely tied to the opiate epidemic. The CDC reports 50,000 Americans died in 2019 from the opiate overdoses, which 25% were physician prescribed. Lives lost and the economic impact drives discovery of safe replacements for opioids as a high priority. Significant economic and societal impact drives the discovery of safe replacements for the 50 million US adults with chronic pain. Chronic pain is responsible for a $635 billion loss to the US economy. This is a bigger economic impact than heart disease, cancer, and diabetes. Dr. Kunamneni at Mayo generates this special type of antibody with ribosome display technology for targets found upregulated in chronic pain. My lab, with 40 years experience, identified the targets, and that of Dr. Sasha Alice at the University of New Mexico. We've moved forward with in vitro and in vivo testing of optimized human variants that reduce action similarly in every chronic pain model we've tested. Preclinical development of humanized small antibodies with current funding can continue through IND-enabling independent confirmations and first-in-man phase trials assisted by milestone-driven NIH cooperative agreements and potentially EPIC-NET funding for phase 2. Our Neurochronics startup company is generating additional funding for CRO validations prior to the phase 1 trials. Our innovation is our small therapeutic biologicals called single-chain fragment variable antibodies, or SCFVs. They're generated with ribosome display technology to bind, in particular, 2 chronic pain mediators, CCKBR and P2X4R. They consist of only the recognition portion of antibodies. And the small VH and VL proteins and their linker are about 30% smaller than the size of a full monoclonal antibody. And because they're small, they have a higher and stronger binding affinity and higher tissue penetration, providing access to pain targets in nerve and in the brain. While this novel concept for pain relief, these other SCFBs have already been approved for other indications, including cancer by the WHO. Thus, our solution for chronic pain with this unique approach is optimized human variants targeting CCKBR and P2X4R, well-known receptors involved in chronic pain. Both candidates provide potent, specific, and durable block for at least 3 weeks in all chronic pain models tested. As a result, anxiety and depression are prevented. Testing indicates that these non-opiates have no no abuse potential, which is further limited since the administration would need to be done in a clinical setting. We're leveraging the ribosome display selection technology into a pipeline of antibodies, in this case for CCK and P2X4 receptors. It's well known that both of these targets and their RNA are involved in chronic pain, anxiety, and depression. They remain upregulated, and this is key, for up to 5 times in chronic pain models, although their presence is normally lower. High binding affinity and the selectivity of the engineered biologicals is shown by the low nanomolar KDs, which indicate superior binding affinity that has improved with the humanization of the variants compared to the parent mouse SCFB. An effective 50-fold dose has a favorable safety profile in vitro and in vivo. The in vivo test with a facial trigeminal nerve injury chronic pain model indicate the biologics are safe and have potent efficacy. The blue timelines are the sensitivity levels in naive animals. When the models are induced with the nerve injury, the sensitivity reaches a low plateau at 3 weeks, shown by the red line. When we treat with the SCFBs at 3 weeks, the chronic pain sensitivity is greatly reduced back toward baseline. The reduced sensitivity level is equivalent to the standard of care shown by the green line, which only lasts 4 to 12 hours, depending on the formulation. But the effects of the SCFB are durable. And the NC002 P2X4 SCFB continues on up to baseline. Similarly, the biologics reverse both sciatic nerve foot hypersensitivity, and in an inflammatory back pain model, the recovery is back to baseline. Both the SCFBs reduce anxiety induced by chronic pain in a dose-dependent manner in the models, and preventing it at the higher dose. With in vivo whole cell patch clamp recordings in mouse neurons, a much lower rate of neuronal firing is shown after direct application of the CCK-BR SCFB onto neurons isolated from the ganglia of the mice from the facial pain nerve injured animals. The CCK receptors on neurons activated with the agonist CCK8 are even further amplifies the SCFB effect of the treatment. It supports our behavioral data. Now, the preliminary data that we have that's most exciting is that when we apply the SCFB directly onto human neurons, isolated from donations of the donor service, there is a greatly reduced frequency of firing in the patch clamp recordings. And more current is required to apply to these neurons to get them to fire after the treatment with our SCFB. This directly validates more clearly the potential of the humanized SCFBs for human use, and motivates us to move forward toward IND filing and phase 1 trials that can be funded partially through the NIH programs. We have acquired national and international intellectual property filings and have published the mouse antibody studies. The Neurochronic startup company will move to lead these biologics forward toward IND filing. The scientific team includes myself and Dr. Alves at the University of New Mexico, along with Dr. Kunamneni at Mayo Clinic, who generates the SCFBs with his technology. Dr. Asanian coordinates with the donor team to provide the human neurons for testing, and he will be involved in the clinical trials as he has done at Stanford. Dr. Ada Santiago is a successful pharma independent consultant brought in by the management team of Suman Law, CEO of Neurochronics, and Bill Chen, board of directors. All have developed many pharmaceutical products brought into market, particularly Dr. Chin. They will be leading the validations forward with investments that supplement the additional NIH funds toward phase trials. So we have a novel approach to pain management, which is in use for other conditions such as cancer, Huntington's, and Parkinson's. We've identified at least 2 targets upregulated in chronic pain, which is a new strategy with the RNAs that persist long-term in the injured animals. And with the humanized small antibodies, we have durable efficacy in numerous chronic pain models and in human neurons preliminarily now. In addition to the $3 million that we've already garnered in federal grant funding, potential funds from NIH and investor support will move to lead the candidate forward toward IND filing and phase trials to reach our goal of generating effective, durable, non-narcotic chronic pain therapy. Thank you to Equalize and Jay Lichter, my mentor, who has helped me to make great strides forward. Thank you.
Thank you so much, Karin. Meghana, would you like to make the first question or comment?
Yeah, just curious. I mean, you mentioned these 2 targets upregulated in chronic pain. Just out of curiosity, what's sort of the normal function of these targets?
The CCK-B receptor is very much involved in anxiety. In human studies many, many years ago, these have been studied a long time ago, but no therapy has come forward, probably due to specificity issues. But if CCK, the peptide, is injected ICV into a human brain, there's a clinical report saying that a great anxiety event occurs. So it's well known in the higher brain levels that the CCK is involved in anxiety in particular. And the CCK is located throughout the pain system. It's a copeptide in many of the pain neurons and the facilitatory pain pathways. The P2X4 is it's one of the P2X4, there's many. There are many others that are present in normally. This particular one is sequestered in endosomes near the membrane. And when the neuronal activity begins, it pops out of the endosomes and into the membrane. So my key strategy has been to focus on things that are normally lowish, and then are suddenly upregulated with nerve injuries and inflammatory pain conditions, and they're maintained at higher levels. And many of the early pain studies, we know that many pain drugs have failed. But most of the pain research studies done for decades have involved acute pain models. And I use models that are not so harmful to the animal, but they persist up to 100 days that I've looked. So this has been our strategy.
Great. And just obviously this isn't a narcotic, but you presume that it will target the problem of addiction with opioids.
Yes, we need alternates desperately.
Great.
So we'll need more studies, more CRO studies to verify what I have found about that. It's non-narcotic, but that it won't be addictive like
Right.
some of the current new neuropathic pain treatments have become addictive.
All right, thanks, Meghana. Chris Brunner.
Yeah, this is a very nice presentation. Maybe a little bit of an orthogonal question. You've got multiple potential assets here that are based on this technology. How are you thinking about prioritization of your efforts going forward? And then separately, beyond chronic pain, as you think about other potential utilizations for the SCFVs, how are you kind of thinking about that longer term?
Okay, so the first question is, we're right now deciding, and within the last few weeks, I've been very much leaning toward the CCK SCFV, which is why I presented more of that data. And my rationale is that it not only works with chronic pain, but it's known historically in the literature to be involved with anxiety. Therefore, it may have more general use. Also, the P2X4, and this is well known in the literature, is a mechanism that is only viable to block in male mice in this case. The female involves T cells and peripheral. And we've tried many, the females in all of our models, and this particular SCFB is not effective in female animals. So the CCKB seems to be a more useful product overall. And we know that we've recently started doing multiple dosing. And with multiple dosing, like once a week, we can get much, much closer to the naive baseline.
Thanks, helpful.
Chris Garabedian.
Sure. Yeah, really nice presentation, very comprehensive, and you hit on a lot of key things. So actually, you just hit on my one of my questions was the dosing interval and how you can, because it looks like all the animal work has been done with a single dose. So better understanding a strategy around that, especially if it's competing for more chronic administered drugs. But just a comment, you framed it through the opioid, and that's been a really big challenge. But nociceptive pain and neuropathic pain is a very clear dividing line for the most part. And
Yes.
while there are opioids used for neuropathic pain, I think better elucidating the neuropathic pathway, which has had their own list of failures,
Yes.
and how you might compare to the gabapentinoids, right, which are approved and generally used first line. So I'm just curious, are there any models where the gabapentinoids have performed well, where you could use that as a positive control and compare your drugs to? And then just so if you can answer that, but the other comment is your budget. I just commented that $1 million was light for IND enabling for a small molecule. I think 10 to 15 million feels light for 2 antibodies with manufacturing, IND enabling work, and getting through a phase 1. And so really, are you thinking about just parallel tracking both of these products? Or are you thinking that you need better elucidation of the properties to decide which one is the lead horse?
Well, that's where the experience that I've gotten from Jay Lecter is that realistically only one can go forward. But the neurochronics team and Bill Chin and Suman Lal will be very much a part of that. And we're still at the decision point of which way to go with these, because it would be unrealistic probably to take both of them forward.
Yeah, and just a question about the gabapentinoid comparison in a preclinical. Are there any models you could run these products against?
Models, I've done 4 or 5 models in animals. We do use the gabapentinoids as control comparisons. And the fact is that they come up and they go down, the pain is up and it's down. And with this, although it takes a few weeks for it to happen, it's just such a durable effect that I think it's an entirely different class, a different approach. There's always the danger, as there is with gabapentinoids, that it will diminish in effectiveness. It will become habit forming, apparently. And so since there's nothing quite like this, I can't really make a comparison.
Yeah, I think you're answering my question, which is that would be very compelling data. If you had data to show that these are clearly differentiated from a class that's used widely for neuropathic pain, that if that came out and you had good data, I would bring that forward. But Karin, nice presentation. Thank you very much.
Yeah, but we do have in the papers of the mice, we have some RNA-seq data, and we're waiting some more that shows that the values are down, the RNAs are back down. And so it looks like a true reversal of function and settling back down to naive levels.